Assalamu alaikum. The lecture of this today is about Vibrio. Vibrio is the genus of gram-negative bacteria. Contain many species, but the three species are important are Vibrio cholera, which causes cholera disease. Vibrio parahemolyticus cause gastroenteritis, wound infection, and bacteremia. Vibrio vulnivacus cause wound infection and bacteremia. First, I'm talking about Vibrio cholera. Vibrio cholera is the causative agent of cholera disease. Cholera disease is an acute diarrheal disease characterized by massive loss of body fluids, watery diarrhea, vomiting, thirst, dehydration, and muscle cramps. The patient can be deadly within hours if not adequately treated. This disease is induced by enterotoxin, and this disease is non-invasive diarrhea disease because the organism not reach the blood and act locally. Vibrio cholera has over 150 identified serogroups, but the two main serogroups based on O antigen, which is somatic antigen O1 and O139. These two strains or these two serogroups are toxigenic and cause cholera disease. O1 has two biotypes, classical and altur. The altur is a predominant cholera pathogen worldwide. راح أشرح لكم هنا لماذا سميت بالتور. تور هو الاسم الذي يطلق على السلالة المعينة من البكتيريا ضمن الفيبريو كوليرا. تم التعرف على هاي السلالة في عام 1905 في مخيم الحجر الصحي في شبه جزيرة سيناء في الطور في مصر من قبل الطبيب الألماني جوشليش، حيث تم العثور على هذه السلالة في الفيبريو كوليرا في أمعاء 6 من الحجاج العائدين من مكة المكرمة، على الرغم من أن الحجاج لم تظهر عليهم أي أعراض أو علامات سريرية قبل الوفاة، فلهذا سميت بالتور. التور يعني الطور. Well, the important properties of this bacteria is coma-shaped, so it is called Vibrio, gram-negative aerobic bacillus, contain two antigenic structures, H flagella and O antigen, somatic antigen, contain lipopolysaccharide in the cell wall, oxidase positive, and it is actively motile via polar flagellum. Well, modes of transmission. This bacteria grow normally in salt and fresh water and can survive and multiply in water by infecting poppy pod. So the modes of transmissions are unsafe water identified this as a major route, unwashed fruit and vegetables or washed in bad water, left over rice not are heated, lack of hand washing, food preparation, hand shaking, cooked and uncooked seafood, shellfish, inadequate sewage treatment, and lack of water treatment. From person to person, direct from person to person is very rare. I say in the start of this lecture, only serogroup O1 and O139 are toxigenic and cause cholera disease. Why? Because these two serogroups contain many virulence factors. This play a major role in the pathogenesis. The important one is cholera toxin. The function of this, or the biological effect of this virulence factor, is hypersecretion of electrolytes and water. Cholera toxin or enterotoxin or cholera gene, many names of this toxin, a protein that causes profuse watery diarrhea, and this diarrhea known as rice water stool. Well, another virulence factor is coagulated pilus adherence to mucosal cells is the function of this virulence factor. Also it is contain accessory colonization. The virulence factor hemagglutination important in induce intestinal inflammation, while protease or mucinase, the function is degradation of the tight junction. So mucinase or protease, degradation the tight junction. Siderophores, it is the iron carrier. Siderophores are small high-affinity iron compounds that are secreted by microorganisms, and the function is transport the iron to the cell or bacterial cell across the cell membrane. Finally, neuraminidase, very important for increase toxin receptors, because this toxin binding to the intestinal cell through the receptors. Well, as I say, the virulence factor cholera toxin cause hypersecretion of electrolytes and water. Well, so the Vibrio cholera produce the toxin called cholera toxin or enterotoxin, which have 5 pentamer subunits called B. 1, 2, 3, 4, 5. 5 molecule or 5 pentamer of the B subunits, and A subunit which contain 2 molecule A and 2 linked with the other. B subunits helping to attach to the ganglioside or G protein, so the toxin enter. When the toxin enter, the A cleaved into the A1 and A2. A1 very important because it is responsible for the enzymatic activity, and R increased the adenylate cyclase activity. So it is increased the cyclic AMP of intestinal cell. As a result, it allows the massive loss of water as well as the ions sodium, chloride, potassium, and bicarbonates. The release of the different ions as well as the water, so the cell lack everything which is needed or requirement for growth, nutrition, and development, so it causes diarrhea. If the patient not treated immediately, it finally die in hours. this bacteria cause infection. Let me talking about the pathogenesis of this bacteria. So cholera caused by the enterotoxin that promote the secretion of fluid and electrolytes into the lumen of the gut, and the result in watery diarrhea with the electrolytes concentration isotonic to those of plasma. And this toxin or enterotoxin act locally and do not invade the intestinal wall. So this microorganism cannot reach to the blood. Well, the fluid in the duodenum, and in the upper jejunum is lost, but ileum not affected. The colon is a state of absorption because it is not sensitive to the toxin. So the large volume of fluid produced in the upper intestine overwhelm the capacity of the lower bowel, which result in severe diarrhea. Laboratory diagnosis. The sample is stool because this bacteria causes watery diarrhea. Staining by gram stain, it appears as a gram-negative common shape bacilli. Well, culturing, the selective and differential culture, it is called TCPS. It is the abbreviation of the thiosulfate citrate bile salt saccharose. So the Vibrio cholera ferment the saccharose and appear as golden or yellow colonies, while the Vibrio parahemolyticus cannot ferment the saccharose and appear as dark green. So this yellow colony, it is Vibrio cholera, while the dark green, it is Vibrio parahemolyticus. The other lab finding: incomplete blood count, increased white BC, hematocrit, blood urea, and serum creatinine. This is because the hemoconcentration, because the dehydration and bicarbonate loss in stool leads to the metabolic acidosis. Total body potassium is depleted. Well, now I'm talking about Vibrio parahemolyticus, another species of the Vibrio. This species require for the NaCl 3.5% NaCl for culturing, and it is called halophilic Vibrio. Also, it is this bacteria or this species has the hemolysin virulence factor related to its pathogenicity, can detected by human or rabbit RBC test. The disease is food poisoning in human, and this bacteria transmitted by raw sea food. This bacteria gave the same morphology as Vibrio cholera, curved gram-negative bacilli, facultative anaerobe, but requirement to the salt 3.5% for growth, and the virulence factor are hemolysin and adhesion. Because they are ranging from mild to cholera-like disease, and the typical The presentation of this disease is an explosive watery diarrhea and less commonly associated with the wound infection and bacteremia. The diagnosis the same in the Vibrio cholera by culturing on TCP as. Thank you.